Hello, everyone. I am義守University School of Medicine, Liu Lifen. Today, I would like to introduce the heritage and gene and the application. In this class, we will begin with the basic concept of heredity, how traits are transmitted from parents to offspring. Then we move into gene, chromosome, gene expression, and several common genetic diseases. In the second half of the lecture, we will discuss how genetic change contributes to cancer development. And finally, we will introduce the major application of genetic engineering in medicine, agriculture, livestock, aquaculture, food production, and the chemical industry. The goal of this lecture is not only to understand genetic concepts, but also to connect them with real clinical disease and biotechnology application. This slide shows the structure of today's lectures. There are 6 major parts. First, we will introduce heredity, including its definition and the scope of genetic research. Second, we will discuss genes, including their structure, function, and how genes are expressed. Third, we will introduce chromosome and the concept of allele. Fourth, we will look at the disease-related genes and how they are mapped on human chromosome. Fifth, we will discuss several common genetic diseases such as albinism, thalassemia, spinal muscular atrophy, and the G6PD deficiency. Finally, we will talk about cancer and genetic engineering, including how mutation cause cancer and how genetic engineering is applied in modern biotechnology. By the end of this lecture, students should be able to explain the relationship among DNA, gene, chromosome, traits, inherited disease, cancer, and the biotechnology application. Let us begin with the concept of heredity. Heredity refers to the process by which offspring acquire traits from their parents through the transmission of genes. In other words, biological information is passed from one generation to the next. This process allows parental trait to be maintained and continue in offspring. The core mechanism behind heredity is the replication and the transfer of genetic information. In most live organisms, this genetic information is stored in DNA. DNA contains the instruction necessary for growth, development, function, and reproduction. The scientific discipline that studies heredity is called genetics. Genetics examines gene structure, gene function, genetic variation, and inheritance pattern. Although nearly all known life forms use DNA as their genetic material, some viruses use RNA instead. This reminds us that heredity is a universal biological concept, but the molecular forms of genetic information may vary among different biological systems. Now we are moving from heredity to the basic unit of heredity, the gene. A gene is a functional sequence of DNA that carry genetic information. It considers the basic heredity unit. They control traits. Genes do not directly produce traits by themselves. Instead, the information encoded in gene is expressed through a series of molecular processes. First, DNA is transcribed into messenger RNA. Then messenger RNA is translated into protein. This protein performs biological function and contributes to observable characteristics, which we call trait. This process is known as the central dogma of molecular biology. Human has over 20,000 to 25,000 genes. These genes are distributed across 23 pairs of chromosomes and together form the human genome. Therefore, when we study genes, we are studying the molecular instructions that help determine how the human body development, function, and response to gene disease. The development of organism from a single fertilized egg into complete body is one of the most remarkable biological processes. This process requires precise regulation of gene expression. Cells must know when to divide, when to differentiate, where to migrate, and what function of the perform. This regulation occurs through gene expression cascade. A gene expression cascade means that protein produces one group of gene activating another group of gene. This sequential activation allows development to proceed in an organism manner. An important example is the homeotic gene. Homeotic genes are the master regulator gene. They determine the identity of body segment. If the gene mutates, body parts may develop in the wrong location. For example, in classical development biology study, legs may grow where antennae should be. In addition to gene cascade, cells must communicate with another. This is called cell signal. Cell release signal molecules, and the other cell receive the signal through receptor. Through this communication, cells coordinate their behavior during embryo development and throughout adult life. This slide explains how cells receive and respond to external signal. A signal transduction pathway is the process by which an external signal is received at the cell surface and transmitted into the cell, eventually leading to a specific cellular response. The process begins when a signaling cell releases signaling molecules. These molecules may be hormone, cytokine, growth factor, or other small molecules. Next, the signaling molecule binds to receptor protein on the surface of a target cell. This binding activates the receptor. After receptor activation, intracellular relay protein transmits the signal step by step inside the cell. This relay protein often form a cascade event and amplify the signal. Eventually, the signal activates transcription factor. The transcription factor enters the nucleus and regulates gene expression. As a result, specific genes are turned on or turned off, leading to protein production and the cellular response. This mechanism is essential for development, tissue repair, immune response, and the normal physiological regulation. However, abnormal signal transduction is closely associated with disease, such as cancer and autoimmune disorder. Here, we summarize the 6 key steps of signal transduction. First, a signal cell secretes signal molecules, also called ligand. Second, the ligand binds to a receptor protein on the target cell. Third, receptor activation triggers a cascade of relay protein inside the cell. Fourth, the signal is passed from one molecule to another until transcription factors are activated. Fifth, the activate transcription factor enter the nucleus and promote transcription of DNA into RNA. 6. The mRNA is translated at ribosome into new protein. This newly synthesized protein can carry out specific cellular function. The important point is the signal transduction connects the external environment to gene expression inside the nucleus. Any disruption in this pathway may lead to abnormal gene expression. For example, if a growth signal is always activated, the cell may divide continuously. This is one of the key mechanisms involved in cancer development. Next, we introduce chromosomes. Chromosomes are highly organized structures made by DNA wrapped around histone protein. They become especially visible during cell division when DNA is tightly condensed. In human somatic cells, chromosomes exist in pairs. Human has 23 pairs of chromosomes for a total of 46 chromosomes. One chromosome in each pair is inherited from the mother, and the other is inherited from the father. Each chromosome carries many genes, and each gene is located at a specific position. To bring us to the concept of allele, allele is different version of a gene located corresponding position on homologous chromosome. These alleles control the same trait. Alleles can be dominant or recessive. A dominant allele can produce a trait even when only one copy is present. A recessive allele usually requires 2 copies for the trait to be expressed. This concept is fundamental for understanding inheritance patterns such as autosomal recessive and X-linked inheritance. Although almost all cells in the body contain the same genome, but not all genes are expressed in every cell. For example, a neuron, a muscle cell, and a pancreatic beta cell all contain the same DNA. However, they look different and perform different functions because they express different sets of genes. Pancreatic islet cell express genes related to insulin production. Neuron express genes involved in neuron transmission. Muscle cells express genes such as actin and myosin. They are important for contraction. This is called cell-specific gene expression. Gene expression must be precisely controlled. If a gene that should be activated is deleted, mutated, or silenced, disease may occur. On this slide, chromosome 1 is shown with several disease-related genes, such as GBA associated with Kucher's disease, and the PS2 gene is associated with Alzheimer's disease. This example shows the genetic diseases are often linked to specific genes located on specific chromosomes. This slide shows examples of disease-related genes located on chromosome 2 to chromosome 9. For example, chromosome 2 contains genes such as MSH2 and NSH6, which are associated with colon cancer. These genes are involved in DNA mismatch repair. When they are defective, mutation accumulation more easily. Chromosome 3 contains the VHL gene associated with von Hippel-Lindau disease, and the MLH1, another important mismatch repair gene related to colon cancer. Chromosome 4 contains the HD gene associated with Huntington disease, and the FGFR3 associated with achondroplasia. We will discuss achondroplasia later. Chromosome 5 contains SMN1, which is associated with spinal muscular atrophy. The key message is the human chromosomes are not only structural carriers of DNA, They also present a map of disease-related genes. By locating genes on chromosomes, doctor and the researcher can better understand inheritance, diagnosis, and the disease mechanism. This slide continues the mapping of disease-related genes on chromosome 10 to 17. These chromosomes, including genes related to metabolic disease, neurological disease, endocrine disorder, and cancer. For example, chromosome 6 contains genes associated with spinocerebellar ataxia, hemochromatosis, diabetes, congenital adrenal hyperplasia, and epilepsy. Other chromosomes in this range contains important cancer-related genes and the gene involved in inheritance syndrome. What is the mapping important? In clinical medicine, if a patient has a suspected inheritance disease, genetic testing can identify mutation in specific gene. This allows doctors to confirm diagnosis, predict disease risk, provide genetic consulting, and sometimes select target treatment. Therefore, chromosome mapping connects basic genetics with clinical practice. This slide shows disease-related genes on chromosome 18, 22, X, and Y. The sex chromosomes are especially important in inheritance. Female usually have 2 X chromosomes, while male usually have 1 X and 1 Y chromosome. Because male have only 1 X chromosome, mutations in X-linked genes are more likely to cause disease in male. This is why many X-linked recessive disorders such as G6PD deficiency and hemophilia occurs more frequently in male. The Y chromosome carries genes important for male sex determination and fertility. This slide reminds us that location of gene affects not only the disease mechanism, but also the inheritance pattern and the risk among male and female offspring. Now we will move from gene map to specific genetic disease. In this section, we will discuss several common and clinical important genetic disorders. These include albinism, achondroplasia, anemia, spinal muscular atrophy, and G6PD deficiency. For each disease, we will focus on 3 major points. First, what is the inheritance pattern? Second, what is the pathogenic mechanism? Third, what are the major clinical features of daily life precaution? Understanding this disease helps students connect molecular genetics with real clinical presentation. Achondroplasia is autosomal dominant disorder that is the most common forms of disproportional short statures. It is mainly caused by mutation in FGFR3 gene located on chromosome 4. FGFR3 stands for fibroblast growth factor receptor 3. Normally, FGFR3 helps regulate bone growth. However, in achondroplasia, the mutation causes abnormal activation of FGFR3. This inhibits the proliferation and the differentiation of chondrocytes, which are cartilage-forming cells. As a result, long bone growth is impaired. Clinically, patients have short limbs with related normal trunk. They may have an enlarged head size and abnormal front head and mind-face hypoplasia. Importantly, intellectual development is generally normal. Although achondroplasia is autosomal dominant, about 80% of cases occurs due to new mutation, also called de novo mutation. This means neither parents have the disease, but the mutation rests spontaneous in the child. Thalassemia is a congenital autosomal recessive disorder affecting hemoglobin production. Hemoglobin is an oxygen-carrying protein in red blood cell. It is composed of globin chain and heme groups. In thalassemia, mutation or deletion impair the production of globin chains. As a result, a red blood cell becomes smaller, more fragile, and have a short lifespan. This leads to chronic hemolytic anemia. There are two major types, alpha thalassemia and beta thalassemia. Clinically, patients may present with anemia, fatigue, pallor, jaundice, enlarged spleen, and several cases, growth retardation, and bone deformation. In Taiwan, the carrier rate is relatively high, estimated at around 5 to 10%. Therefore, thalassemia is an important target for gene screening. The disease demonstrates the importance of carrier screening in public health and preventive medicine. Spinal muscular atrophy (SMA) is an autosomal recessive disorder. It is primarily caused by deletion or mutation of SMN1 gene on chromosome 5. SMN1 gene is essential for survival of motor neuron. When SMN1 gene function is lost, anterior horn motor neuron in the spinal cord progressively degenerate. These neurons are responsible for controlling muscle movement. As they are lost, patients develop progressive muscle weakness and atrophy. One impaired feature of SMA is that intellectual development is not affected. Patients may have normal cognitions, even when motor function is severely impaired. SMA is classified into 1 to 5 based on the age of onset and clinical severity. Type 1 is the most severe and usually begins in infancy. SMA is therefore an excellent example of how understanding gene disease can lead to advanced molecular therapy. G6PD deficiency, also known as Bevins, is an X-linked recessive disorder. G6PD stands for glucose 6-phosphate dehydrogenase. This enzyme is important for maintaining antioxidant capacity of red blood cells. Red blood cells are consistently exposed to oxidative stress. G6PD helps produce NADPH, which maintains glutathione in its reduction form. Reductive glutathione protects red blood cells from oxidative damage. When G6PD activity is deficient, red blood cells become damaged to the oxidative injury. Certain triggers cause red blood cell destruction, leads to acute hemolytic anemia such as drug and fava beans. Because the X-linked recessive males are more commonly affected than female, patients should avoid known triggers and seek medicine attention if they develop jaundice, dark red urine, tea-colored urine, severe fatigue, or signs of hemolysis. This disease is also clinically important for newborn screening and medication safety. Now we move to the next major topic, the origin of cancer. Cancer is not usually caused by a single event. It is a complex disease driven by accumulation of genetic mutation and epigenetic change. These changes affect genes that regulate cell growth, cell division, DNA repair, apoptosis, angiogenesis, invasion, and metastasis. Understanding the genetic base of cancer is essential for prevention, diagnosis, prognosis, and treatment. In the following slide, we will discuss how normal cells become cancer cells, what types of genes are involved, and why cancer development usually acquires multiple genetic changes over many years. Cancer begins when genes are controlled. They control cell division become abnormal. Normally, cell division is tightly regulated. Cells divide only when needed, and the damaged cell may stop dividing or undergo apoptosis. However, when mutation occurs in genes controlling growth, division, cell may contribute dividing without normal regulation. This can lead to tumor formation. Tumor can be benign or malignant. Benign tumor usually grows locally and do not invade surrounding tissue. Malignant tumors are invaded and can spread into distant organs. This process is called metastasis, and the malignant tumors are what we commonly call cancer. Many factors can increase cancer risk. This including cigarette smoking, poor diet, UV irradiation, alcohol consumption, ionization irradiation, chemical pollutants, viral infection, and inhaled chemical. A key concept is the proto-oncogene. Proto-oncogenes are normal genes. They promote cell growth. When properly regulated, if they are mutated, amplification, or abnormal activate, they can become oncogene and drive uncontrolled cell dividing. Some viruses can also contribute to cancer. For example, HPV virus is associated with cervical cancer, and HPV is associated with liver cancer. Cancer development usually requires multiple genetic changes. There are two major categories of cancer-related genes: oncogene and the tumor suppressor gene. Oncogene act as an accelerator. When activated, they promote cell proliferation. Tumor suppressor gene acts as a brakes. They suppress abnormal cell division, repair DNA damage, and promote apoptosis. When tumor suppressor genes are mutated or inactivated, the brake is lost. Well-known tumor suppressor genes including p53, BRCA1, BRCA2, and the RB gene. Cancer usually requires more than one mutation. In many cases, at least one oncogene must be activated, and the tumor suppressor pathway must be inactivated. Because multiple mutations are required, cancer often develops over years or decades. This is why prevention and early detection are so important. Avoiding tobacco, reducing exposure to carcinogen, maintaining a healthy lifestyle, and receiving regular screening can significantly reduce cancer risk and delete cancer earlier. This slide shows how proto-oncogene can become oncogene. There are several major mechanisms. First, a point mutation may alter the protein structure, make it continuously activate. For example, mutation in ROS gene can keep growth signaling pathway turned on. Second, gene amplification may increase the number of copies of the proto-oncogene. More copies lead to overproduction of growth-promoting protein. Third, chromosome translocation may move a proto-oncogene next to a highly activated promoter or fuse with another gene. This can result in abnormal expression or production of a fusion protein. Fourth, viral insertion may disrupt normal gene regulation. Viral DNA inserted into the host genome can activate oncogene or interferes with tumor suppressor genes. All these mechanisms can convert a normal regulated gene into cancer-promoting oncogene. The important concept is the oncogene usually act as gain-of-function manner. One abnormal copy may be enough to promote cancer development. Tumor suppressor genes function like the brakes of cell division. They help prevent uncontrolled growth by regulating cell cycle, repair DNA damage, and including apoptosis when damage is too severe. The two-hit hypothesis explains how tumor suppressor genes are inactivated. Because human has two copies of most genes, both allele of tumor suppressor gene usually needs to be mutated or lose functions before the protective function is completely removed. In inherited cancer, a person may inherit one defect copy from the birth, and this is the first hit. Later in life, if the second copy is lost or mutated in a somatic cell, the second hit occurs and the cancer risk arises. This explains why hereditary cancer often occurs younger age and may affect multiple family members. In contrast, sporadic cancer requires both hits to occur during a person's lifetime in the same cell lineage. This concept is very important for understanding inherited cancer, susceptibility, genetic consulting, and the cancer screening strategy. Colon cancer is the classic example of stepwise cancer development through accumulation of mutation. This process often begins in a normal epithelial cell of the colon. The first key events may be mutation of APC gene, leading to forming a small polyp. APC is a tumor suppressor gene involving in regulating cell growth. Next, mutation in RAS gene may promote further proliferation, leading to a medium-sized adenoma. Later, inactivate a gene such as DCC or SMAD4 may contribute to the development of large adenoma. Finally, mutation of P53 and the additional genetic change may drive progression from adenoma to invasive carcinoma. This process usually takes 10 to 20 years. This long time window provides an important opportunity for early detection. Colonoscopy can identify and remove precancer polyps before they become invasive cancer. Therefore, colorectal cancer is an excellent example of how understanding cancer genetics directly supports cancer prevention. This slide summarizes the major biological change that occurs as normal cells become cancer cells. First, cells acquire unlimited proliferative capacity. They divide more than they should. Second, they escape apoptosis. Normally, damaged cells undergo programmed cell death, but cancer often avoid this mechanism. Third, tumor develop angiogenesis, which means they stimulate new blood vessel formation. Blood vessels provide oxygen and nutrients to support tumor growth. Fourth, cancer acquires the ability to invade surrounding tissue. Finally, they metastasis to distant organs through blood vessel and the lymphatic vessel. This process is not sudden. It is a gradual evolutionary process. Mutation accumulation and cells with growth advantage are selected over time. This is why cancer is both genetic disease and an evolutionary disease at the cellular level. Understanding this process helps explain why cancer treatment often requires combination therapy and why early detection is critical. Now we enter the final part of today's lecture, genetic engineering and its application. Genetic engineering has transformed modern biotechnology. Genetic engineering, by directly manipulating genetic material, scientists can modify organisms to produce useful protein, improve crops, generate vaccine, create disease model, and develop advanced therapies. The impact of genetic engineering extends across medicine, agriculture, livestock, aquaculture, food production, and the chemistry industry. In the following slide, we will look at several important examples that show how genetic knowledge can be translated into real-world products and technologies. Genetic engineering refers to a group of biotechnology techniques that directly manipulate an organism's genome. It is also called genetic technology, gene transfer, genetic modification. The core conception is to introduce, remove, and modify genes in a target organism so that organism gain a new trait or produce a specific product. One of the earliest and the most important applications of genetic engineering was the production of protein-based drugs. Before genetic engineering, many therapeutic proteins had been extracted from human or animals' tissue. This made them scarce, expensive, and sometimes unsafe. For example, including human growth hormone, insulin, and interferon. With genetic engineering, human genes can be inserted into microorganisms such as bacteria or yeast. These organisms can act as biofactories to produce human proteins in a large quantities. The completely changed the production model of the modern medicine. One of the most famous examples is insulin production. In the past, insulin was extracted from the pancreatics of cows or pigs. One patient's annual insulin requirement could require many animals. This was inefficient, expensive, and sometimes caused immune response. With genetic engineering, the human insulin gene can be inserted into bacteria, such as E. coli. These bacteria can produce human insulin rapidly and in large amounts. The greatly reduced cost and improved drug availability. Another example is growth hormone. Traditionally, human growth hormones are extracted from human tissue, which was extremely limited and carried safety concerns. Genetic engineering made recombinant growth hormone production possible. Similarly, small peptide hormones such as somatostatin also can be produced using engineering microorganisms. The key message is the genetic engineering changed rare biological molecules into scalable medical products. Genetic engineering has many medical applications. The first example is insulin production. Genetic engineering E. coli can mass production human insulin, which is now widely used for treating diabetes. The second example is the hepatitis B vaccine. Genetical engineering yeast can produce hepatitis B surface antigen. This recombinant protein is used to make a safe and effective vaccine. The third example is the production of therapeutic protein in transgenic animals. For example, sheep can be engineered to express human alpha-1 antitrypsin in their milk. After purification, this protein can be used to treat patients with alpha-1 antitrypsin deficiency and related lung disease. This example demonstrates that genetic engineering can be used not only to produce drugs, but also to prevent infectious disease and treat inheritance protein deficiency. Genetic engineering is also widely used in agriculture. One application is in improved nutritional value. For example, rice can be modified to increase essential amino acid content, such as lysine. Golden rice is another example. It contains gene to produce more vitamin A precursor. This is particularly important in regions where vitamin A deficiency causes blindness and immune problems. A second application is to improve disease resistance. For example, introducing viral cold protein genes into plants can make them resistant to certain viral infections. Similar approach can help cross resistant bacteria, fungi, and other pathogens. A third application is to reducing pest damage. Bt crops contains gene from Bacillus thuringiensis. This gene encodes Bt toxin protein. They are toxic to certain insect larvae. As a result, the crops are protected from the pest damage and may require less chemical pesticide. Overall, agricultural gene engineering can improve yield, quality, sustainability, and food security. Genetic engineering is also used in livestock and agriculture. By introducing growth-related genes, animals such as pig and salmon may grow faster, use feed more efficiently, and reach market size sooner. For example, transgenic pigs carry growth hormone-related genes may show accelerate growth, improve feed efficiency, and reduce fat content. Transgenic salmon are another well-known case. By modifying growth hormone regulation, this salmon can grow faster and larger than the conventional salmon. Commercialized product has already entered the market in some regions. Another interesting example is fluorescence fish. These fish carry fluorescence proteins such as GFP gene. They grow under specific light source. Besides their commercialized value, fluorescence proteins are also widely used in biochemical research to trace gene expression and cell behavior. This example shows that genetic engineering can affect not only medicine but also food production, animal breeding, and research tools. Genetical engineering microorganisms are widely used in the food industry. They can produce enzymes, amino acid, organic acid, vitamin, pigments, flavoring compounds, and functional protein. For example, chymosin is used in cheese production. Traditionally, it was extracted from cow's stomach. Today, recombinant chymotrypsin can be produced by microorganisms, improving consistency and reducing reliance on animal resources. Genetic engineering is also important in the chemical industry. Engineering microorganisms can produce enzymes such as protease, lipase, cellulose, and amylase. Protease help break down the protein stains in laundry detergent. Lipase help remove oil. Cellulose can use in texture process. Amylase can be used in food process and paper manufacturing. These biochemical enzymes support the idea of green chemistry because they can reduce the use of hazard chemicals, lower pollution, and improve industry efficiency. Thus, genetic engineering has become a key technology in sustainable biotechnology. To conclude today's lecture, we have discussed heredity, gene, chromosome, genetic disease, cancer genetics, and genetic engineering application. We begin with the idea that heredity allows genetic information to pass from parents to offspring. We then learn that genes are DNA sequences that encode functional products, and the gene expression determines many cellular traits. We also discuss chromosome and allele, and we connect specific gene to inheritance disease. In the clinical genetics sections, we review albinism, achondroplasia, thalassemia, SMA, G6PD deficiency. This example shows how inheritance pattern and the gene mutations explain disease mechanism. In the cancer section, we learned that cancer development through the accumulation of multiple genetic changes involving oncogene and the tumor suppressor gene. Finally, we discussed genetic engineering and its application in medicine, agriculture, livestock, aquaculture, food production, and the chemical industry. The major take-home message is that genetics is not only a basic biological science, it is also fundamental for modern medicine, biotechnology, disease prevention, and future therapeutic innovation. Thank you for your attention.